Welcome to your first video lecture on Human Anatomy 2. And our first chapter is chapter 17 of your Human Anatomy textbook by Marieb and Katja Hohen. And we're going to get into the blood. The blood is the life-sustaining transport vehicle of the cardiovascular system. It delivers oxygen and nutrients to body cells, transporting metabolic waste to lungs and kidneys for elimination, transporting hormones from the endocrine system to the target organs. And pretty much it has other functions such as regulation function, which includes maintaining body temperature by absorbing and distributing heat, maintaining normal pH using buffers. It's also an alkaline reserve of the bicarbonate ions, maintaining adequate fluid volumes in the circulatory system, and pretty much also for protection, the body protection. It prevents blood loss, and the plasma proteins and platelets in the blood can initiate clot formation. Preventing infection also is one of them, and agents of immunity are carried in the blood. You have your antibodies and a complement system, the white blood cells. Um, um, a lot of things are actually dissolved in the blood, which is actually for our protection. So it is indeed a life sustaining and as well as transport system of the body. So the transport function includes delivering various substances throughout the body. Regulation function, just like what I mentioned, maintains body temperature, maintaining normal pH, maintaining adequate fluid volume, and then the protection includes preventing blood loss and preventing infection. Quite clear. So let's take a look at what the blood is made of. The blood is the only fluid tissue in the body. It is a connective tissue. The matrix is non-living fluid called the plasma, and you can see here, this is whole blood in this picture right here. This is whole blood that's been placed in a centrifuge, and then the centrifuge spins the blood, separating it into its most dense component and its least dense component. So the most dense component of the blood stays at the bottom, and that's actually your erythrocytes. And then the least dense is actually the plasma, which is actually nothing but water and some dissolved proteins in it. But there is something like a very fine line right there, which we call the buffy coat. And that buffy coat is also filled with formed elements that are less dense than the red blood cells. So all of this is actually suspended in the plasma. Let's take a look at more of its physical characteristics as well as volume. Blood is sticky and opaque fluid with metallic taste. Color varies with its oxygen content. A high oxygen content would show a very bright red or scarlet red blood, and the low oxygen content would be showing a dark red, almost brown blood. It has a pH of 7.35 to 7.45. It makes up about 8% of the body weight, and the average volume among males will be between 5 to 6 liters, and then the females being like on average we are smaller, between 4 to 5 liters. So let's take a look at the components of the blood. When you separate blood, it is separated into its cellular components and also the fluid component, which is actually the blood plasma. The blood plasma is stroke colored, yellowish stroke colored, and you could see right there in the picture that's actually real blood plasma. And it's kind of like a sticky fluid. It has over 100 dissolved substances, pretty much nutrients, gases, hormones, proteins, inorganic ions, and the proteins albumin, globulin, and fibrinogen is actually also in your plasma. So albumin makes up about 60%. This part here, which is, let me see if I could turn on my pointer. This part here, which is albumin, takes up about 60% of the plasma protein. And its function is to carry, it's a carrier molecule, and it's also a blood buffer, and it contributes to the osmotic pressure of the blood. The reason why the reason why we are keeping blood inside our body is because it is being kept in by the oncotic pressure of the protein albumin. And here you are, that's actually your plasma protein. We've discussed already the albumin. Let's get into globulin. The globulins pretty much would be less, and it is actually made up of the alpha, beta, and gamma globulins, which are only a few of the many subtypes of proteins that are more complicated, really, than albumin. We have the alpha globulin, which is a group of protein um, that is called the alpha fetoprotein, which is generated by the fetal liver and is utilized as a marker for certain cancers. And uh, pretty much we have also one that is really most common among the globulins is actually the immunoglobulins. The immunoglobulins is commonly referred to as the gamma globulins, and these gamma globulins is an essential component of our immunological response, which means our immune system makes use of these immunoglobulins. As a matter of fact, we call them antibodies. Sometimes they get interchanged when you say immunoglobulins, you are also talking about antibodies. But remember this, guys, all antibodies are immunoglobulins, but not all immunoglobulins are antibodies. So there are certain immunoglobulins, certain classes of immunoglobulins that are not necessarily called or is not necessarily acting as antibodies. But all antibodies are regarded as immunoglobulins. And finally, the last one is actually a complex protein, which is called fibrinogen, that is involved in the clotting process. And there are other proteins besides the fibrinogen and the globulin and the albumin that is part of the plasma, but they are found in smaller quantities, such as your lipoprotein, which transports lipids throughout the body. You have also the enzymes that is actually acting as metabolic workers of the body, which would help in speeding up chemical reactions. They are catalysts. So they're all in the plasma. And here is actually a composition of the plasma table, which will help you in your review. This is in your textbook or something that would look as close as this table here. It'll give you an idea of what you find floating in the plasma, and that includes the formed elements, your red blood cell and your white blood cells and your platelets. Then the function of them that are involved there, and then the other parts of the plasma, the other parts of just the plasma with its protein and the function. So pretty much make use of your textbook and read them because they are going to be in your exams. Let's now go to the formed elements, and the formed element. The first one that we're going to have that we're going to do will be to look at the red blood cells. But before we do that, this is already a blood smear right there in front of you. You could see the pink sections. These are your erythrocytes or red blood cells. Those are, and you could see that they do not have any nucleus. Then you have those little dots that are almost as the color of your red blood cell. But remember, the color is actually the stain Usually this is right stain, we call it right stain, and it's being picked up by these formed elements and it's giving them the characteristic color. Like you form the other formed elements, your white blood cells that would have nucleus. These are the true cells, and you could see that they have different shapes of nucleus and they have some granules. Some of them have granules, some of them don't have granules inside. You can see this guy here, which is so beautiful, my favorite. It would have like red granules on them. And I have another favorite that is not really shown here, which is a basophil, unfortunately. But I will show you how it would look like. But then the color of this is not really the color of how they are in the blood. The red blood cells would pretty much like be depending on how much oxygen they're carrying, would pretty much like reflect a crimson color or a red color. But your white blood cells are called white blood cells because they're really white. They only pick up that color once they are already stained, and that's already in the lab. So let's go and take a look at the red blood cells. The red blood cells are small. They're about 7.5 microns in diameter, and they are involved in gas transport. And the cell is biconcave, and you could see it. And the reason why it's biconcave is because the middle part is hollow. It doesn't have any more nucleus, so it pretty much sinks. It is sunken on both sides, so you call it biconcave. And but no, whatever you can feel in this biconcave disc shaped cell pretty much is really nothing but 97% hemoglobin. And the hemoglobin is involved in gas transport. The diameter of the white blood cell is about 7.5 microns, and it contains the plasma membrane. In its plasma membrane contains a protein which you call spectrin, and spectrin provides flexibility to the plasma membrane so that when the red blood cell goes into tight spots, such as the very, very small tight spot, um, what do you call this blood vessel, the capillaries, it could actually like fold and be transported easily right through the capillaries. So red blood cells are dedicated to respiratory gas transport. A hemoglobin reversibly would pretty much like interact with oxygen. So it picks up oxygen has uh, I would not say oxygen, but hemoglobin. Hemoglobin has a very high affinity for oxygen. It has also affinity for carbon dioxide, but between carbon dioxide and oxygen, your hemoglobin loves to pretty much like be in touch with oxygen much more than carbon dioxide. So the normal values of hemoglobin, again, would be different between male and female. Males would have 13 to 18 grams per 100 ml of blood, and your females will have 12 to 16 grams per 100 ml of blood. And that's only the hemoglobin, right? The hemoglobin consists of the red heme pigment that is bound to a protein which you call globin. That's This is a better diagram of your hemoglobin. So hemoglobin is composed of four polypeptide chain. One, two, three, four polypeptide chain. And the uh, the top one here in this diagram is showing two beta globin chains, and the bottom part is actually the alpha globin chains. Whether it's alpha or beta, right in the middle of these globin chains, they have a something that looks like a button, right, with something inside of it. This is your heme group. The heme group is represented here as this red button shaped structures with something red inside. The heme, that little part there on the inside has is actually the part of the heme of hemoglobin that is very much attracted to or has high affinity for oxygen. So oxygen, when oxygen gets loaded in the lungs, it produces oxyhemoglobin that makes the blood like ruby red or really very, very bright red. Now, when you unload the hemoglobin, when we exhale, or when you unload oxygen in the tissues, because oxygen is carried to the tissues because we need oxygen in the tissues. But once it is unloaded there, the hemoglobin now becomes what you call oxyhemoglobin or reduced hemoglobin. And this entire hemoglobin, when it uploads its own oxygen, would probably upload only probably like one oxygen for every how many millions of white red blood cells we have. Uploading one will is enough for us really to breathe, and our tissues to be breathing and taking up what we breathe. Trust me. So it is uploading the oxygen and then loading in the tissues, loading in carbon dioxide. 20% of the carbon dioxide in the blood would bind to hemoglobin, producing a complex unit which you call carbaminohemoglobin. Erythrocytes are being produced in the bone marrow in a process which you call hemopoiesis. It is Hemopoiesis is all general processes involved in the formation of all blood cells, and it occurs in the bone marrow, the red bone marrow, which is composed of reticular connective tissue and blood sinusoids. In adult, it is found in the axial skeleton, the girdles, and the proximal epiphysis of the humerus and the femur. I'm hoping that you still remember those structures in your coming from your AMP1. Now, the great great grandmother of all blood cells, whether you're talking of red, white, or even the platelets, is a um, is a cell which is called the hemocytoblast. This is a stem cell, the hemopoietic stem cell called hemocytoblast, give rise to all formed elements, hormones, and growth factors. They push the cell towards a specific pathway of blood development, and the committed cells cannot change back anymore. They will need to go forward. You cannot go backwards. We need to go forward into their destination, into their maturity. They either become, because the hemocytoblast can be an red blood cell. A committed red blood cell becomes a red blood cell. A committed white blood cell becomes a white blood cell, and a committed platelet from a megakaryocyte, the committed platelets, the megakaryocyte will burst, and all of those remnants of the bursting of your megakaryocyte will be the platelets. So we have here, we should know also, only on the surface, the stages of erythropoiesis. This is now your erythropoiesis. This is only the production of red blood cell. You have only one great grandmother of all blood cells, the hemocytoblast. It can become a red blood cell, it can become a white blood cell, or a platelet. How will that happen? How can one cell magically transform itself into either one? Well, got something to do with the signaling molecules, which are hormones, will be the one that's going to prod the hemocytoblast to become, depending on what the body needs, to become whatever it is supposed to be. In the case of becoming a red blood cell, a hemocytoblast will need to, in the process of erythropoiesis, would need erythropoietin, EPO, we call it EPO. It is a hormone that would stimulate the hemocytoblast to get into the process and pretty much mature into a red blood cell. And you have here a hemopoietic stem cell, okay, and pretty much it is going to be receiving information from the erythropoietin, and the cell which you call the pro-erythroblast, becomes a committed cell. Once it is committed, it cannot go back to becoming an erythroblast. It has to move forward. It becomes a basophilic erythroblast and then a polychromatic erythroblast into an orthochromatic erythroblast. And all of this will pretty much like lead into the formation of a baby red blood cell right here, which you call a reticulocyte. A reticulocyte has already gotten rid of the nucleus right at this point here where it is in the late orthochromatic erythroblast. your um, your baby red blood cell would have already gotten rid of the nucleus. The nucleus here is not anymore needed because before it becomes an orthochromatic erythroblast, in its prochromatic stage, it is already producing lots of RNA in order that they can produce the protein that is very much needed by a red blood cell, and that is the protein hemoglobin. So this area here, this particular stage right here, is actually where it is actively producing, with the help of its RNA, actively producing protein, or it's actually doing protein synthesis, producing hemoglobin. And once it is done with its job, you won't need the nucleus anymore. You won't need to drag the heavy nucleus in your adult red blood cell stage because you would need to be really running around in the bloodstream and would be very busy picking up substances, and the nucleus is only going to be a heavy load. So the body pretty much like intelligent enough, and we are really intelligently designed, is getting rid of things that will make our what will make our cells imperfect. Although we cannot say that our cells are really perfect, because if our cells are perfect, nobody will die. We still have imperfections, and one of the imperfections of the human body is actually aging, right? We cannot avoid that, but it is what it is. So now we have the baby erythrocytes, your reticulocyte, and your reticulocyte accounts for 1.5% of the blood volume every day. Every day, our hemocytoblast, the great great grandmother, produces 1.5% blood. Why? Why do we have to produce 1.5%? Well, guys, your red blood cell, because it does not have nucleus, will not live long. It lives for about 120 days, and therefore about 1.5% after its 120th day will die also. If it doesn't die and it goes right through the spleen, it will be killed by macrophages, because old red blood cells will become sluggish and will not anymore be doing its job properly, and therefore the macrophages will kill it. 1.5% we kill red blood cells, and then that 1.5% gets replaced in the bone marrow. That's why these baby cells here, which you call reticulocyte, 1.5% of that is always present in our bloodstream every single day. So, hypoxia, too few red blood cells around to do the job would result to hypoxia. Too many red blood cells that would increase viscosity would lead to polycythemia. So there should be a balance between uh, how much red blood cell we're going to have, and the balance is being created by the production, okay, how much is being produced daily, and then how much is lost or how much is actually destroyed. Pretty much about 2 million red blood cells are made per second. With that 1.5% that I'm talking about, 2 million red blood cells are made every second, and there should be a balance with the production and destruction depending on hormonal controls as well as dietary requirements. So the hormonal control is actually the big shot in erythropoiesis, and that is that particular molecule that is capable of telling the hemocytoblast, hey, you have to make red blood cell, and that is your EPO or erythropoietin. And there's always a small amount of EPO in the blood to maintain the basal rate released by the kidneys, okay? And some of them is being released by the liver in order to stimulate the hemocytoblast in producing red blood cells. About 1.5%. If everything is normal and everything is A-okay, it's only going to be 1.5%. So if there is actually like hypoxia, uh, then you are actually assuming that there is actually decreased red blood cells, and a decreased number would lead to less red blood cells that are going to be that are going to be carrying the oxygen around. And if there's going to be low oxygen level, generally decreased number of oxygen in the bloodstream, then you are putting the body in a hypoxic situation. So the reason for this could be insufficient hemoglobin in the red blood cell. Maybe your hemoglobin is very low, or maybe there is a reduced availability of oxygen in your environment. If you are in high altitude, the oxygen level drops. And if you are breathing air that has less oxygen, you are going to be hypoxic also. Or you can have too many erythrocytes for high oxygen levels in the blood, and that too much red blood cell pretty much would go on a feedback loop and would inhibit the release of EPO. Because you have already too much red blood cell, then the feedback loop will be for your red bone marrow to slow down on the production of red blood cell. The EPO, therefore, in general, causes the erythrocytes also not only to multiply in number, but also to mature faster. Another hormone that's going to be affecting EPO, another hormone is testosterone. Men or the males can enhance EPO production through their testosterone, resulting in higher red blood cell count. And that's the reason why they have higher red blood cell count than the females. Now, too many erythrocytes or very high oxygen level would inhibit EPO production, and therefore would, I already said this, would pretty much like as a feedback loop, because too high testosterone will produce more red blood cell. If you're going to have too much red blood cell, there's going to be a feedback loop to reduce the number of EPO, so your red blood cell production is also going to slow down. So here is a feedback loop. I have already mentioned about feedback loop. You should know already the feedback loop in AMP1. You've read about it in the first chapter of your AMP1 textbook. Let's take a look at the EPO mechanism for regulating erythropoiesis. So you have a stimulus. Well, actually, if your C-cell pendant right here is balanced, you have normal blood oxygen level. If you have an imbalance and it tilts to down, that means your stimulus here becomes hypoxia. There's inadequate oxygen delivery due to decreased um, due to decreased oxygen due to decreased oxygen count and decreased amount of hemoglobin and decreased availability of oxygen. So what's going to happen? The EPO, which is produced in the kidneys, and to a small to a small part of it also produced in the liver, will release erythropoietin. Erythropoietin now will stimulate the red bone marrow, and that will enhance erythropoiesis and will increase the red blood cell count. And eventually, it's going to tilt the seesaw balance back into normal by increasing the oxygen carrying ability of the blood. If you have any questions, please make sure that you throw in your questions to me via email on this. So let's take a look at some homeostatic imbalances regarding EPO or the production of red blood cells. Some athletes abuse artificial EPO. So they said, oh, we can actually increase our red blood cell content. Now, athletes, remember, use their muscles, and when you use your muscles, according to AMP1, when your muscles are actually contracting, you are actually using up your oxygen. So that's the reason why when you are doing an exercise, your oxygen level drops and you are going to be breathing faster, and you will actually be in need of oxygen because you are consuming the oxygen in your blood. That oxygen that is supposed to be available to muscles will be reduced as you are overusing your muscles. And athletes are always like that. So, what athletes would want, okay, would be to have a continuous source of oxygen. And the only way to do that will be to increase their red blood cells ability to bring in more oxygen. So their heart rate will actually be uh, pumping and will continually be delivering red blood cells to the muscles that are rich in oxygen. But sometimes that is not enough. You really need a lot of them. So you need a lot more of them by increasing the number of red blood cells, you are going to be pretty much delivering more oxygen, and you are going to have a better muscular control. But it can have dangerous consequences. The use of EPO would always increase the level of red blood cell. It increases hematocrit. It also increases stamina. It improves the performance of the athletes. So the athletes always aim to have more red blood cells to have more of that EPO. So what they do is that there's actually like artificial EPO out there in the market, and they're going to get it and they're going to have an injection of this. EPO can increase hematocrit from 45% up to 65. And you know what happens to the blood when you have a hematocrit of about 65%, having 65% red blood cell in your bloodstream. Pretty much that means that your blood is going to be thick and sludgy and sluggish, therefore. It is going to be very viscous. So this will increase your, dangerously increase your chances of having clotting, thrombosis, stroke, and to the most part, as worse as can be, heart failure or even death. So we can live healthily by just living normally without EPO. We can have like just get our natural daily requirement that is needed by erythropoiesis. So we have proteins, amino acid, lipids, and carbohydrates, most especially iron, because your red blood cell, the hemoglobin, has iron in the middle, and it would need 4 iron for every 1 hemoglobin. And iron in our body is stored as ferritin and hemosiderin, and is transported around the blood from, let's say, the liver or the kidneys to the bone marrow via a protein called transferrin. You always need these proteins to transport iron because elemental iron is very toxic. You cannot have elemental iron floating around in the blood. It has to be, it has to be linked to its protein so that it's not going to be toxic. But of course, you need also vitamin B as well as folic acid, and these are actually um, necessary for DNA synthesis and for rapidly dividing cells, such as your developing red blood cells. The lifespan of erythrocyte is about 100 to 120 days only because of the absence of protein, and it doesn't have much of the organelles inside it. It doesn't have mitochondria. It cannot produce its own. It cannot produce its own ATP, and therefore it dies only. It stays only in our blood for 100 to about 120 days. The old red blood cells, because of the absence of energy, ATP, become very fragile, and it begins to degenerate, and they can be trapped in smaller channels, especially in the spleen, our organ that has a lot of tortuous channel. And the spleen is actually known to be the graveyard of red blood cells, and it has plenty of macrophages, and the macrophages in the spleen can spot a very old and sluggish red blood cell, and they are broken down into its components. So when red blood cells get broken down, they're broken down into its the hemoglobin is broken down into its hemiform and its globin form. They're separated. And let me show you what happens to the fate of your dead red blood cells. Here you have red blood cells, okay? Red blood cells typically would circulate for about 100 to 120 days, would become very old and damaged, and as they age, their cell membrane becomes less flexible. They undergo changes that signal their removal. The macrophages, and wherever they are, most especially in the spleen, will digest them, and they will be broken down into their heme is going well. First, the hemoglobin is going to be broken down into its globin and heme portion. The globin portion in is going to be delivered into the liver and is going to be converted back into new other amino acid that will be used by the body to make our own protein or to make new globin parts for the red blood cells also. Then the heme part, the one that carries your iron, is broken down into the iron part. So the elemental iron gets picked up by a protein transferrin and is delivered into the liver for storage. And also the other part of the heme is called the biliverdin. Biliverdin is converted into bilirubin, and the bilirubin in the liver is from the liver is now going to be channeled into the gallbladder, and in together with the bile inside the bile, it is going to be secreted into the small intestine. In the in it helps one last time in as part of the bile in emulsification of fat that we have eaten in our diet. So the bilirubin is going to be converted into urobilinogen by the bacteria in our small intestine. Some of it is actually going to be some of the urobil some of the urobilinogen is going to be transferred to the liver and converted into urobilin. That is actually not liver, but kidney. And right there in the kidney, it's going to be the urobilin is actually going to be incorporated with the substance that is actually mixed there to become urine, And that's a reason why our urine is actually yellow, because um, bilirubin is actually yellow. Some of them, though, doesn't go to the urine and would become stercobilin in the large intestine and is going to be incorporated as part of the feces. If we eat a lot of fatty food, you will notice that your fecal matter is yellowish, and that's because of the stercobilin. If your fecal matter is not yellowish and is actually dark green or dark brown, that means you do not have much fat to digest, and therefore there's very little of the gallbladder um, secretion that is actually incorporated in your intestine. Remember that the production of the gallbladder on the liver is actually based on your diet, what kind of diet you need. If you need that, then your liver would need to produce more of the bilirubin to be incorporated in the bile. So now let's take a look at the iron part. Iron part, if our bone marrow would need to produce more of the red blood cell, it will call on every parts of the body that to deliver iron to it, and most of the time it's actually the transferrin, the protein transferrin, that picks up the iron that goes to the bone marrow. And once it is this iron now plus the globin that's been digested here is going to produce again your heme and the globin together, and with the help of vitamin B and of course erythropoietin as your um, stimulating hormone, you're now going to be producing or starting erythropoiesis in the red bone marrow. So the cycle again begins. 120 days it's going to live until it dies, and when it dies, it goes back to becoming globin and heme and so on. So obviously, you can see here that we don't really lose iron. We never lose iron. You see that iron goes around. We don't really need to be eating iron. So why is it that our mama is asking us to eat food that's rich in iron? Because it is not always that we don't lose it. If we're not going to be losing whole blood, then we don't need to be taking any more iron. However, most especially among females, our monthly menstrual period, during our monthly menstrual period, we are actually losing whole blood. For males, iron deficiency anemia is a rarity. They're The only time that a male will have iron deficiency anemia is when he's actually losing whole blood. Maybe he has bleeding ulcer, or maybe he had surgery and lost whole blood, or a gunshot wound, or any type of hemorrhage that will actually make him lose whole blood. That's actually going to result to iron deficiency anemia. But otherwise, we really don't lose um, iron except for the females during their menstrual period. So let's take a look at erythrocyte disorders. Most of the erythrocyte disorders are either anemia or polycythemia. Anemia is when the blood has abnormally low oxygen carrying capacity, that is too low to support normal metabolism. Pretty much anemia is pretty much like a sign of a problem rather than a disease itself. The symptoms of anemia will be fatigue, pallor, dyspnea, and chills. And there are three groups of anemia based on the cause of anemia. That is blood loss, or anemia due to blood loss, anemia due to not having enough red blood cell produced, or anemia resulting from destruction of too many red blood cells. Too many, much more than 1.5%. and that will result to anemia. So, anemia due to blood loss is what you call hemorrhagic anemia. And there's also a second anemia that is listed here, which is anemia because there's not enough red blood cell that is produced. Um, the the first one, which is hemorrhagic anemia, is an anemia that is brought about by uh, too much blood loss. Maybe it's because of a bleeding ulcer or anything that is bleeding that can cause you to lose whole blood, and most especially among females, loss of blood during the menstrual cycle, monthly menstrual period. Now, your not enough red blood cell is produced, your iron deficiency and anemia, pretty much is because of uh, low erythropoietin. It's because uh, and the resulting will be if there's not going to be enough hemoglobin that's going to produce, the red blood cells that are produced will be small. They are what you call microcytic. And and pretty much this is exactly how it looks like. Your normal red blood cells are big, this normal size, but your smaller and paler red blood cell has less hemoglobin in it, and therefore you are suffering from iron deficiency. Then you have pernicious anemia, anemia where there's not enough red blood cell that is actually being produced in the bone marrow. Very much, it's an autoimmune. Pernicious anemia is an autoimmune. Um, it's an autoimmune response, and the body is unable to produce enough red blood cell simply because the erythropoietin is not enough erythropoietin is actually being produced to stimulate normal production of red blood cells. It happens, it's an autoimmune problem. So, the next one will be anemia that is brought about again by not enough is being produced, is actually what you call renal anemia. Uh, okay, the problem with not enough red blood cells being produced is always low erythropoietin. You need enough erythropoietin in order to produce enough red blood cell. So in the previous in the previous it looks in the previous slide, we are saying that there's not enough enough erythropoietin that's being produced because it is actually an autoimmune response. So you are really sick, your erythropoietin is not really being released. The next one is because your kidneys are sick. If your kidneys are sick, as in renal anemia, then the kidneys will not be producing enough erythropoietin. This is caused by lack of EPO because of a renal disease. And the treatment here, if you cannot correct the renal disease, will be synthetic EPO. The next one that can bring about not having enough red blood cell being produced is your aplastic anemia. Aplastic anemia is the destruction or inhibition of the red bone marrow. So, if your red bone marrow will be destroyed, it's not going to be able to do its job of producing blood cells. And again, this could either be a type of cancer, okay, or a cancer, or regarded as an autoimmune disorder, where there's actually too much destruction and of red blood cells and inhibition of the red bone marrow. You are definitely not going to be able to produce enough blood to sustain you, to support you. Aplastic anemia is fatal, and the only way that you can get out of this fatal condition will be blood transfusion, or if you're able, or if the doctor is able to correct what is actually causing the destruction and inhibition on the bone marrow itself. So there are actually other disorders that can cause destruction of red blood cell besides aplastic anemia. For instance, you have the hemolytic anemia. Hemolytic anemia can come in as a disorder in the spleen, because in the spleen is the graveyard of all red blood cell. If you have a hyperactive spleen that is causing a lot of red blood cells being killed, then that can actually give you anemia, and that's what you call hemolytic anemia. Next one is actually if you are sick with disorders that can cause destruction of your red blood cells, or that can cause less production of your red blood cells. Examples of this will be your thalassemias, which is a Mediterranean disorder, and sickle cell anemia. In both cases, these are both genetically passed on, and the nature of thalassemia is that you will have small red blood cells. You're going to be producing less hemoglobin in the red blood cell, and if you have, there's actually two major genes that would be two major DNA set that would be carrying thalassemia, thalassemia major, then thalassemia major is actually almost fatal. Yeah, you will need like blood transfusion every 100 days if you will have the major. Thalassemia minor is only carrying the trait. At some point in time, you will live normally for the rest of your life, but you are always anemic. And then sickle cell anemia is again another genetically is actually another genetic blood dyscrasia, and this is because of a mutated hemoglobin, and it's only one mutation of only one nucleotide, and you already have a mutation where the hemoglobin will cause um, the red blood cells to become not its regular shape, and therefore is unable to carry enough oxygen inside it because of its shape, and therefore would render the sick person really anemic. And just like anything else, just like the thalassemia, this will need um, blood transfusion every 120 days. However, there are people who are only carriers. They only carry one gene, only one parent handed down the sickle cell to them, and therefore they are only slightly anemic, and they don't need any blood transfusion. The next disorder, besides anemia, the second disorder is having too much of the blood, which you call polycythemia. Polycythemia means abnormal or excessive red blood cell. And the problem with this, you think it is really probably good because you're going to have lots of oxygen carrier around you, but it'll make your blood so thick, so viscous, and so sluggish that it can lead to a heart attack or clotting or thrombi formation, and pretty much it can actually like be fatal. It can cause death. Long time ago, people who have too much blood, they actually go regularly to the blood bank and give their blood. And long time ago, during the 15th century, they use like um, they seek the help of a nematode to get their blood out of their system, or they give away their blood. That's actually the best thing. Aha, we're now going to the leukocytes, or the white blood cells. White blood cells are only are the only formed elements that are complete. They will have the cell nuclei and other organelles inside them, but they're not really that much. Our blood is red because of the red blood cells. If the white blood cells are going to be much more in number, then our blood will be white, not red. So you know that the blood is made up mostly of red blood cells. Your white blood cells account only for less than 1% of the total blood volume. There's only about 5,000 to 11,000 white blood cell per microliter of blood. For half a drop, about half a drop is 1 microliter. There's about 5,000 to 11,000 white blood cells in it. They function in defense against diseases. They can leave the capillaries, they can walk out of the capillaries and into the tissues via diapedesis. They move through tissue spaces by amoeboid motion, and they has the ability to be attracted to chemicals. It's actually what you call chemotaxis. Your leukocytes are grouped into two major categories. One is called the granular, and the other one is called agranular. But there's actually five leukocytes: neutrophil, lymphocytes, monocytes, eosinophil, and basophil. Matter of fact, according to their abundance in the blood, if I'm going to ask you, and I will be asking you in your exam, to pretty much like put the leukocytes in the order of abundance in the blood from the highest to the most abundant to the least abundant, you have a mnemonic to remember that. Never let monkeys eat bananas. Never is for the N in neutrophil. L is actually for the L in lymphocytes. M is for the M of monocyte. E is for the E in eosinophil. And B is for the B of basophil. This mnemonic is actually accounting for the arrangement of the white blood cell from according to its abundance, from the most abundant to the least abundant. The most abundant being neutrophil. Let's go to the classification of white blood cells. So your granulus, you have two categories by which white blood cells are categorized: the granulars and the agranulars. We're now into the granulars. The granulars are your um, white blood cells that carry cytoplasmic granules. They have granules in the cytoplasm. You have your eosinophil, you have your basophil, you have your neutrophil. They are larger and short-lived than red blood cells, so they can die less than number of days than white blood cells. White blood cells can live up to 100 and 120 days. Your granulocytes live less, depending on how active they are. If you are actually sick and they are actively defending you and they're active in the immune response, they can actually like use up their energy and they can die quickly. Some of them live only a week or sometimes live only like 15 days. They contain lobed rather than circular nuclei. Their cytoplasmic granule specifically would be staining different colors on the right stain. And they are all phagocytic to a certain degree. Right here is your neutrophil. You could see that the neutrophil here is lobed, and each of the subnuclei are actually held together. They are considered to be one nucleus only. If they are connected to one another, they are one nucleus. Then you have a bilobed nucleus here for the eosinophil. But the granule here of the eosinophil is red. This is my favorite white blood cell. I like also the neutrophil, but your your eosinophil is more beautiful. Then rarest of all of them, you have only 1.5% basophil among all of the 1.5% of the white blood cell is basophil. So in a what do you call it, in a differential count of all white blood cell, you are lucky if you find one basophil. And the basophil granules is actually dark stain. Then these two are your agranulocytes. I'm going to get into that when we get into it. Okay, so here's your neutrophil. Okay, the neutrophil pretty much accounts for 50 to 70% of all white blood cells. And pretty much they have the antimicrobial proteins, which are called defensins. They're also known as polymorphonuclear or polys, they call them polys, because their nucleus is lobular. Their nucleus could be anywhere from 3 lobes to about looking like grapes, 6 lobes. There's your neutrophil right there. These neutrophils are known to be very phagocytic, and they're also known to be like bacteria slayer. They actually kill microbes by giving them what you call respiratory burst. Then you have my beautiful eosinophil. They account only for about 3%, an average of 3% of all nucleosides. Their nucleus is lobed usually, and they have red cytoplasmic granules. They resemble earmuffs. Earmuffs with a very nice red dotted hat. They release enzymes on large parasitic worms. Therefore, they are. And is it good for dewormer? No, they're not. They're not really deworming it. But if you have worms inside your blood, not inside your gut, but your blood, then your white blood cell, the eosinophil, is going to take care of that. They play major role. They pretty much like multiply when you have allergies, when you have asthma. They're actually into the active mode of immune response. The basophil, the last of the granular leukocytes, is the rarest and accounting only for about 5.5% to 1% of leukocytes. They're large, they're almost like purple black. They have granules that contain lots of histamine, and they are functionally similar to mast cells. Histamine, if you are pretty much like, if you're going to go back into tissue repair, I think chapter 4 of in your AMP1, pretty much histamine is a chemical that of inflammation. It acts as a vasodilator and would attract white blood cell to the inflamed area. Functionally, the job of your basophil, because they produce histamine, is just like the mast cell. Remember, your mast cell produce histamine. Now let's get into the agranulocytes. These white blood cells lack visible cytoplasmic granules, and there are two types, the lymphocytes and the monocytes. The lymphocyte, as the second most numerous white blood cell, accounts for about 25% of the white blood cell population. They are large. They are not really large. They are, as a matter of fact, the lymphocytes are the smallest of all, smallest in size. They're so tiny of all the leukocytes. But tiny as they have large dark purple nuclei, circular nuclei, and you could see very thin rim only of the cytoplasm, but you cannot see any granules around. That's why you call them agranulocyte. And there are two types of lymphocytes, the lymphocyte T The lymphocyte T act against various infected cells and tumor cells, and then the B lymphocyte. The lymphocyte T is actually produced in the thymus gland, and then the lymphocyte B is produced in the bone marrow and gives rise to plasma cells, which produce antibodies. So between the two types of lymphocytes, the antibody producers are your B cells or lymphocyte B. Then you have the monocyte. They are big, the largest of all the leukocyte, and they account only for about 3 to 8% of all white blood cells. They can leave circulation, enter the tissue, and they can become a macrophage. These are actively phagocytic cells. They're very voracious. They eat almost like anything that they can see that needs to be cleaned up and eaten, put away, they will do that. And they are crucial in protecting the body against viruses or intracellular bacterial parasites and chronic infections. So, we are going to briefly and zoom right into leukopoiesis. Leukopoiesis is the production of white blood cells and are stimulated by two types of chemical messengers. We are not talking here anymore of the erythropoietin. Your hemocytoblast is still the great great grandmother. Even the leukocytes come from the hemocytoblast. But this time, if the hemocytoblast is not stimulated by erythropoietin and is stimulated by interleukins or colony stimulating factors, they are not going to become red blood cells. They will become white blood cells. All leukocytes originate also from the same great grandmother cell, and they branch into two pathways, the lymphoid stem cell and the myeloid stem cell. And you have two, well, instead of EPO, you have the interleukins, which is usually numbered by, if my memory serves me right, interleukin 3 and interleukin 5. Then you have the colony stimulating factor, which is named after the white blood cell they stimulate, such as, you will have the CSF, but if you are stimulating or producing granulocyte, then you will have granulocyte stimulating factor, okay? Or if you are actually uh, if you are going to stimulate the lymphoid stem cell, then you are going to have the colony, what do you call this? The lymphocyte colony stimulating factor. So let's take a look at the production itself. So your the hemocytoblast gets to be stimulated, okay, and the myeloid stem cell is now going to be the precursor that's going to be producing. And from this myeloid stem cell, which is already committed or slightly committed, it will again be stimulated by colony stimulating factor to be specifically uh, specializing or specifically getting into its more specific leukocyte. You have either a myeloblast, okay, um, these are already committed cells, and that would become, let's say, either an eosinophil, a basophil, or a neutrophil. And these are already the mature granulocytes. The last of the other myeloblasts would could actually become a monocyte. Monocyte is also coming from the myeloid stem cell. There's only one type of white blood cell, the lymphocyte, that do not come from the myeloid stem cell, which comes from the lymphoid stem cells. And these are your lymphocyte, the B cell and the T cell, but they all come from the hemocytoblast. Let's take a look at some homeostatic imbalance. The clinical use of the uh, uh, the hormones, that is for erythropoiesis, for leukopoiesis, is actually for cancer cells that are receiving chemotherapy or that would need stem cell transplant. For these stem cells to produce the type of blood that they need to produce, then these particular hormones are being injected to them. And also for AIDS patient, they are used to increase protective immune responses of AIDS patients. So let's take a look at some disorders of the white blood cell. An overproduction, abnormally high overproduction of white blood cell can cause leukemia. There's two of them, leukemia and infectious mononucleosis. Although leukemia can be drug induced, most particularly it can be drug induced if you are actually like receiving anti-cancer drug or glucocorticoids. Removing those anti-cancer drug can actually bring your white blood cell count back to normal. However, if not, then you have really what do you call this, a disorder that is brought about by abnormally low production of leukocyte, or abnormally high, so rather high production of white blood cell. Then you have uh, leukemia. And leukemia, if not corrected, is fatal. It's actually a cancerous condition and is brought about by overproduction of the abnormal white blood cell. And when you have a lot of white blood cell and they are voracious eaters, they're macrophages, they can kill your own red blood cell. And usually these leukemias are named according to the abnormal white blood cell clone that is involved. They are very acute, they're quickly advancing because they're derived from the stem cell. But they're also like the slow advancing because it involves proliferation of the later cell stages. But either way, it's fatal. If not, really like halted, it's actually fatal. Infectious mononucleosis is also known as the kissing disease. It is highly contagious. You get it by uh, physical touch, kissing. That's why they call it the kissing disease. It's caused by a virus, the Epstein-Barr virus. It results in high numbers of typical agranulocytes. The symptoms, the symptoms would be like the symptoms of having flu. So lethargy, tiredness, achy body, sore throat, low fever. They usually run course with rest within 4 to 6 weeks. You get over it, but you can get it again. Once you have this infectious mononucleosis, you can get it again. When your resistance is low, you get it back. Now we have the platelets. So the platelets, like your red blood cell, are not normal cells. They are fragments of cells. And pretty much they come from very large cells that divide many times, but they don't really undergo cytoplasmic division. The only thing that divides inside the cell are the nucleus. So there's going to be nuclear divisions for about 4 or 6 times. So now you have a large cell that would have a lot of nucleus inside, and it becomes so big that it cannot hold itself and it will burst. then it will spread all of those purple staining granules, which you call platelets. So platelets are nothing but fragments of a larger megakaryocyte. And circulating platelets are always inactive because they're kept inactive by nitric oxide and prostacyclin, which is a substance that is actually produced by the endothelial cells lining the blood vessels. So the platelets are happy and they're not bursting, they're not really causing any problem because of this nitric oxide and this prostacyclin. However, once something happens that is going to bring about the reduction of nitric oxide and prostacyclin, then your platelets will burst. Let's find out what's going to do that. First, the formation of platelet is brought about by another hormone which you call thrombopoietin. And it is formed in a myeloid line from megakaryoblast and would become a large cell called a megakaryocyte. But platelets age and quickly degenerate in about 10 days. So we produce platelets every day, almost every day. About 150 to 400,000 platelets per ml of blood must be with us to be normal. So again, this is in your textbook, a summary of formed elements of the blood. You have the illustration of the blood cell, the description, and then the amount in the blood, the duration in the and lifespan in the body, and the function. This is a very good summary of the anatomy of the blood. Now we're going to go into, we're getting close to the end. But first, let me just take a break and take something so rest my voice for a while. And let me just stop. Okay, pause this. What you have here now is hemostasis. Hemostasis or stoppage of bleeding. What happens when we bleed? What will the body do in order to stop the flow or the escape of that vital fluid, which we call the blood? The body will do everything in its capacity to stop the destruction of blood, to stop also the escape of blood, and pretty much because the blood is a vital component of the human body. So we're going to get back to this, and pretty much at the end of this game, we're going to see this same picture again. Okay, that will first give you, showing you the injury to the blood vessel wall and the escape of red blood cell, which pretty much would result into first vasoconstriction, that particular part where there was actually a cut is going to actually do vasoconstriction. But vasoconstriction is only good for the little blood vessels, most especially the one blood vessels close to the skin, the capillaries and everything. If your major blood vessel, if you get stabbed and major blood vessel will be cut, the vasoconstriction of that major blood vessel is not going to help in the stoppage of bleeding. So this is good only for small blood vessels. The first reaction will be vasoconstriction that will prevent further escape of blood, and then activation of the platelets so that the platelets can actually form a plug right there on that area to avoid further loss of blood. And then after that will be a cascade of chemical reaction in the blood. It gets to be stimulated and it will result, the end result, and there's actually like several control mechanism, antithrombic control mechanism that would lead into the stoppage of this reaction right here. Because once coagulation, what you call this, once there's actually coagulation cascade, thrombin is going to be formed, and then that thrombin is going to be getting it, will be converting fibrinogen into fibrin, and that fibrin is actually the clot. It together with the platelet plug, it is actually going to prevent further escape of blood or further blood loss. That's actually the way it is. Now, in order to stop this, you have antithrombic control mechanism. That's actually in a feedback loop in order to uh, degrade the clot that's been formed and allow tissue repair. This clot cannot stay there. It has to be degraded, it has to be digested, um, and pretty much in order for new tissues to take over that particular spot where the clot is formed. So let's take a look at hemostasis. So hemostasis is a very fast series of reaction for stoppage of bleeding. It requires clotting factors and substances that's released by the platelets. A first step is actually vascular spasm. It's like what I mentioned to you, the vascular spasm, and then formation of platelet plug, and then finally the formation of fibrin through a cascade of mechanism which we call blood clotting. Okay, so vascular spasm happens when the blood vessels get to be damaged. When it gets to be damaged, there's going to be a feedback loop that's going to be sent to the muscles of the small blood vessels, and it will make it constrict. Then platelet plug is going to be formed whenever there's actually like collagen that's been exposed. That's going to pretty much like uh, the the mechanism that's going to start the platelet plug formation. The platelets will adhere to the collagen. A lot of it will explode and will give away or will produce sticky substances, and more of them will pile up and and they will get to plug that particular opening there. And finally, blood coagulation would stop the flow of blood in that particular area. Okay, the main thing in platelet plug formation is a formation of what they call the von Willebrand factor. The von Willebrand factor will help stabilize the platelet collagen adhesion. Your platelet will adhere to the collagen itself because of that factor right there. So you need collagen. You need the collagen in the tissue of the blood vessel to be exposed in order for the von Willebrand factor to be produced. So the von Willebrand factor is like a glue. So that glue is going to be right there, and that glue is going to invite the platelets to stick to it. So when activated, the platelets would swell, would become spiked and sticky, and they would release chemical messengers. ADP causes more platelets to stick and and release their contents. And serotonin and thromboxane A would enhance vascular spasm and platelet aggregation. These are all being produced at the point of injury. The third one is coagulation or blood coagulation, which is a pathway. It's actually two pathways where the end result will be the production of what we call a prothrombin activator, right there, prothrombin activator. So you have the intrinsic and the extrinsic pathway. Pretty much the extrinsic pathway is where the vessel endothelium would rupture and would expose underlying tissue and the collagen itself. The platelets will cling Platelets will cling in their surfaces and provide size for mobilization of these factors. I am not going to ask you to memorize the cascade of these factors, okay? But what I want you to know that the extrinsic factor happens in the endothelium of the blood vessel. The extrinsic pathway, on the other hand, this happens inside the blood itself. This one, the extrinsic pathway, happens outside of the blood. This is happening inside the tissue. The tissue cell trauma exposes the blood to tissue factor, and that tissue factor will be the one that is going to cause a cascade of chemical reaction that eventually will also result in the production of prothrombin activator. Whichever way, this one is longer and this one is shorter, whichever way it will go, inside the blood or in the tissues, both of them will result in the production of prothrombin activator. Once you have the prothrombin, once you have the prothrombin, this pathway will now be stimulating the conversion of prothrombin. The prothrombin activator will activate the conversion of prothrombin to thrombin. And thrombin would catalyze the conversion of fibrinogen into the fibrin clot. This is very important. Thrombin is the one that would make or that would create or that would result in the conversion of fibrinogen to the clot itself. There you go. We are back to the component, the diagram that when I started hemostasis. Here, when you have the fibrinogen converted into fibrin and you form a clot, the clot cannot stay there. The clot will have to be dissolved so that it will be replaced by normal tissues. So you have an antithrombic control mechanism that would prevent this thrombin from continuously being, being produced and continuously forming clot. You cannot allow clot to pretty much like overwhelm a particular reaction. This is a positive feedback, but once you have already the clot, it must stop. There should be fibrinolysis and clot degradation afterwards in order for tissue repair um, to start. And this is now where we have to get in. Clot retraction. Clot retraction is the consolidation or tightening of a clot. When there is already a clot, it doesn't stop right there. Your clot needs to tighten up. It's just like a net, a fisherman's net that's actually holding the hole around in order that the hole is not going to let the blood escape. And that particular net is going to tighten. Fibrin threads in this particular part, fibrin threads will contract and shrink, and it will pull the edges of the vessel wall together, making sure that the repair is going on. And this is actually going to make the repair faster, tighter, and stronger clot formation is happening. Now, the timing here is that the reaction will have to occur and be over and done with. Clot retraction will have to be over and done with within 30 minutes to an hour. So your vessel is actually healing even as clot retraction is occurring. There's actually a substance called PDGF or platelet derived growth factor that is released by the platelets. It stimulates the division of smooth muscle cells and fibroblasts to rebuild blood vessel wall. Then you have also the vascular endothelial growth factor, VEGF, and it stimulates the endothelial cells to multiply and restore endothelial lining. Fibrinolysis is actually the process where clots are removed after the repair is completed. It begins usually within 2 days and continues for several days until the clot is dissolved. A substance called plasminogen, a plasma protein that is trapped within the clot, is converted to plasmin. Plasmin is a fibrin digesting enzyme. A TPA, or a tissue plasminogen activator, as well as factor 7 and thrombin, all play a role in the conversion process. So now we're going to get into the disorders of hemostasis. Of course, our body wants to stop bleeding, but then there are things that the body may encounter that will not allow hemostasis to occur. For instance, if you have impaired liver function, if you have hemophilia, or if you have disseminated intravascular coagulation, pretty much you're not going to be able to stop you're not be able to start hemostasis. Impaired liver function would disable you to synthesize the clotting factors, including vitamin K, and deficiency of this will actually result to impaired hemostasis. And this could be because probably the person may have hepatitis or cirrhosis of the liver, any disease of the liver that would disable that would disable the liver from producing the anticoagulant factor that it needs to produce. Hemophilia is actually a hereditary bleeding disorder. It's pretty much like um, a monosomal. It's actually monosomal. It's um it's inherited from the mother, and it's actually passed on. It's passed on to the mother, and most of the time, the one that is it's an X-linked trait, and therefore the one that is uh, affected most of the time is actually the male, the male child. Then you have your disseminated or disseminated intravascular coagulation. Oh, before I do that, let me give you what the symptoms are of hemophilia. Hemophilia will pretty much like the symptoms would include prolonged bleeding, most especially into the joint cavities. And the treatment of this is pretty much a genetically engineered, um, well, actually genetically engineered factors that um, would make the liver produce the antihemophilic factor. Then you have your DIC, or disseminated intravascular coagulation. It involves both widespread clotting and severe bleeding, and it occurs in an intact blood vessel, blocking blood flow and severe, pretty much it can cause severe bleeding. It can occur in disorders, if you get this kind of disorders like septicemia, or pretty much incompatible blood transfusion, or pretty much complications in pregnancy. So this ends our, no, I'm not going to end it because I still have blood typing, blood transfusion. Okay. So what happens to the body when you lose blood? Well, when you lose blood and the body is unable to stop you losing the blood, and there's nothing you could do, then you have to go and take measure on getting new blood for yourself. If it is actually an emergency and you lost a lot of blood, then blood needs to be transfused. But blood transfusion is not is easily said than done. And you do blood transfusion only if it is necessary. Loss of about 15 to 30% can cause pallor and weakness. Loss of more than 30% can result in potentially fatal severe shock. So, when you need blood, you have to receive blood. But in order for us to receive blood, we have to understand that each of us have different blood types. The humans actually have different blood groups. We have the red blood cells, human red blood cells, and these are the human red blood cells. The plasma membrane of the human red blood cells bear specific antigenic markers. Red blood cell antigens are referred to as agglutinogens because they promote a reaction which you call agglutination. Agglutination is the clumping of red blood cells. When red blood cells clump together, they're not supposed to be clumping, they're supposed to be free flowing into the blood vessel so that they can freely flow into the tissues and give oxygen to the tissues. And together with the other substances of the blood, they need to go through the capillaries and into the tissue to deliver to deliver the things that the tissue needs. If the blood cells are clumped together and they're holding on to each other, that is not going to happen. So you avoid clumping. However, if your blood has been mismatched, you have been transfused with blood that is not the same as your blood type, that can actually cause agglutination or clumping of your red blood cell. How is agglutination happening? Well, first of all, know that each blood type cell, is actually different from one another. You have the group A, they all have antigens or agglutinogens in their red blood cells, in the cover of their red blood cells. That would be agglutinogen A. In the group B, or blood type B, the plasma membrane of these red blood cells have glutinogens that are called agglutinogen B. And then in the group AB, both agglutinogen A and agglutinogen B are considered to be present in the group AB. However, the group O, or blood type O, do not carry any agglutinogen in their plasma membrane. They have no agglutinogen at all. So, besides the agglutinogen, there's another thing that pretty much like coexist with the groups. Now, besides the antigen, there's actually what you call the antibodies. Antibodies are present with the agglutinogens. And what are antibodies? Well, antibodies are substances that pretty much would um, agglutinate the uh, carriers of antigens that is actually a mismatch to them. For instance, antibody B will agglutinate any red blood cell that would have agglutinate B or antigen B. The antibody A will agglutinate any red blood cell that would carry an agglutinogen A or antigen A. So anti-A is against A, anti-B is against B. That's why you don't find them together. Anti-A, you will find them only coexisting with B. It will not be agglutinating B. It will not react with B because it's anti-A. It's against A. On the other hand, A, you will not find A with anti-A. You will find A with anti-B. They coexist together, they're nice together. But what's the purpose of an anti-B with the A? Well, anti-B will prevent the wrong transfusion, possible mismatch transfusion of a B into a blood type A. So a blood type A will pretty much have coexisting with it an antibody B. That prevents a B from being mistakenly transfused into it. Once a B gets into the body of a blood type A, then the antibody will agglutinate that B. The body will not, the person will have a reaction and would not accept that B. Meanwhile, you have the group AB. The group AB has both the antibody, or both the agglutinogen A and B, but doesn't have, it's not showing any antibodies. You cannot have any antibodies with a blood type AB because AB has agglutinogen or antigen A and B. Meanwhile, blood type O does not carry any agglutinogen in its plasma membrane, but coexisting with it is actually in the blood, in the plasma, is actually the antibody A and antibody B. It will have antibody A and antibody B. It prevents any blood bearing agglutinogen A or agglutinogen B from being mistakenly transfused or mismatched into blood type O. They will definitely not be received properly. Besides the blood type A and B, or the ABO system, we call that ABO system, we have also the RH factor. Let me see. Do we discuss the H factor now? I don't think so. Uh, oh, I need to mention the blood antibodies, the anti-B and anti-A. They are also known as agglutinins. You might see the word agglutinin instead of antibody in your exam. Just make sure that you know agglutinins are actually the ones that are preformed with the antibodies that are preformed with the blood type. Agglutinin A is an antibody preformed with blood type B. Antibody B is the agglutinin that is preformed with blood type A. Then you have the other group, the second group, which you call the RH blood group. The RH blood group pretty much has 52 named RH agglutinogens. It's very complicated and it's not necessarily like taken into consideration unless the person really needs like a pretty much like an overhaul of its blood, meaning the entire blood needs to be removed and put together again. Then you need really to have the uh, the RH compatibility determined because there are 52 named RH agglutinogens and they need to be together, or an organ transplant. An organ transplant, if the person is going to have an organ transplant, they need to consider which particular RH group you are referring. But the difference between the RH groups is actually like minimal and could be considered insignificant when you are only going to be transfusing, let's say, a pint of blood. It doesn't really matter. You say RH positive, you don't really need to find out which of the 52 agglutinogens make you positive. You can get any RH positive blood. But if you're going to have organ transplant, you need to know which of the 52. You need to know a little bit of, you need to be a little bit more specific. Among the 52, the RHC, D, and E are the most common. There is antibody against RH positiveness. You call it the D. Okay? This is not D antigen, D antibody. And 85% of the Americans are actually RH positive. I am RH positive. My blood type is A positive. Anti-RH antibodies are not spontaneously formed in Rh negative individuals. So if you are an Rh negative individual, and if you are a woman and you have not had any child yet, you have to make sure that you know if you are an Rh positive or Rh negative individual. Because Rh and negative antibodies are not formed spontaneously in Rh negative individuals. These antibodies are formed, unlike the ABO system, the antibody B for the Rh are not spontaneously formed in Rh negative individuals. They are only formed through second exposure to Rh positive blood. And typically, this will result in a typical transfusion reaction after that. Something is actually going on, or something may happen for among females when females get pregnant and they are Rh negative. And the babies could actually suffer from hemolytic disease of the newborn, otherwise known as your erythroblastosis fetalis. And this one only occurs among Rh negative mom with Rh positive fetus. Now let's take a look during the first pregnancy. During the first pregnancy, this mother here, who is Rh negative, had an Rh positive baby inside her. And during the first, second, and third trimester of pregnancy, there's going to be slight uh, exposure of the mother. Mother will be exposed to the blood of the fetus. Some blood can actually get into the mother, and pretty much it exposes the mother, who is Rh negative, to the baby. The mother will now start learning how to produce the antibody against the Rh positive. But it is too late after when Rh negative, when Rh antibodies are formed in the mother, the baby by the time would have been born. The second time, though, that the mother gets pregnant after that first exposure, the second time that the mother gets pregnant, the mother knows already how to form these antibodies. If there's going to be another growing fetus that is Rh positive in the mother's womb, during the first and second trimester, if there's going to be mixing of the fetal blood into the mother, that will immediately, because the mother knows already, she has learned already how to produce the antibodies, she will immediately produce a lots of antibodies, and these antibodies can now be transferred into the fetus and can kill the fetus. That particular death of the fetus because of Rh incompatibility is what you call erythroblastosis fetalis. How do you stop this? Can you stop this? If you're not going to stop this, then all Rh negative mother will only have one child and they will not anymore have any child. All of the next child will be dead. They will all have erythroblastosis fetalis. A long time ago, those things happened. Not anymore now. Today, when you get into your doctor for prenatal uh, blood, um, you know that you're pregnant. Your doctor pretty much first thing that the doctor will do as part of the examination will be to find out your blood type. If you are Rh negative, the doctor will ask you, have you been exposed previously to, let's say, blood transfusion? Or have you been pregnant before? If you say, no, I have not been pregnant, I did not have any blood transfusion, you think your doctor will stop there and will not do anything anymore? No, because you might have had a previous miscarriage that you do not know or you did not know. There are some women who can actually be pregnant for 1 or 2 months and they did not know it and they would lose the pregnancy and they did not even know they got pregnant. If that pregnancy is Rh positive and you're Rh negative, you have already been exposed. So a good obstetrician will not take things for granted. Immediately, when a mother is Rh negative, the doctor will give the mother an RHOD. This is actually a type of immunoglobulin. This erythroblastosis fetalis can be prevented by desensitizing all Rh negative mothers by giving them injections of massive doses of anti-Rh antibodies, which you call the Rho D immunoglobulin. After every abortion, miscarriage, or even delivery of an Rh negative, if the person is Rh negative, they are not going to assume if the baby is actually positive or negative. They will need to make sure that if the mother has been sensitized, and she may have been sensitized, you can lose your baby, your next baby. These antibodies bind to and inactivate the fetal Rh antigens on the fetal red cells that are present in maternal circulation. In this way, the Rh antigens from the mother's blood are cleared and removed before they have had the time to stimulate production of Rh antibodies, which means they're not going to be putting in any active Rh antibody into the system of the fetus to kill it. So, transfusion reactions would happen if there are actually a mismatch. Usually, it will not happen if there's a good pre-testing of the blood, and a comprehensive pre-testing of the blood will avoid mismatching. Just know in your exam, you may be asked, blood type O is the universal donor. Okay, it will not have A or B antigens or anti-agglutinogens. Blood type AB is the universal recipient, and it will not have antibody A or antibody B. Take a look at this because this is going to be in your lab. I know you don't have any lab, but this is going to be presented in your lab. Just know that agglutination, this is real human blood. When you put antibody A in a drop of blood and all of a sudden it looks like this, usually the blood looks like this. When you put antibody A and it turns like this, that means the red blood cells clump together. So an antibody A will agglutinate a blood that would have antigen A. So a blood that has antigen A is blood type A. So you know that if you put antibody A in a blood and it becomes this, then the blood is actually going to be or would have antigen A. If there's going to be reaction in also the antigen B, and if you put antibody B into this blood and it had a reaction, agglutination, then both A and B are present. So that's blood type AB. If there's no reaction in B and there's a reaction in A, so this is only blood type A. If there's a reaction in B and no reaction in A, so this is your blood type B. Blood type A will be like this. There won't be any clumping or agglutination. How do you know if it is positive or negative? If it is positive or negative, you have another spot in the middle where you put antibody D, anti-D, which is the antibody for Rh. If it agglutinates, then it is Rh positive. If it did not agglutinate, then it is Rh negative. Please make sure that you learn the diagnostic blood test. This is actually in your blood in your lab. There's your differential white blood cell count. Pretty much it would look at relative proportions of each white blood cell. Now the differential white blood cell count will help the clinician determine the type of infection that you are actually having. If you, let's say you have a proliferation of eosinophil, because eosinophil is actually uh, in charge of uh, immunologic reaction during asthma attack, or pretty much like um, parasitic infection, then you know that it could be like an asthma attack or a parasitic infection. It's not only asthma attack really for eosinophil. If you have large amount or large number of eosinophil, could be any allergic reaction that will be increasing the number of eosinophil. So a differential white blood cell count can help determine your what your disorder is. Prothrombin time and platelet count will assess the speed of your hemostasis. A CMP, or a comprehensive medical panel, is a blood chemistry profile that checks various chemical levels that would pretty much like point to any disorder either in your liver or in your kidneys. A complete blood count would check your number of formed elements. It will also check on your hematocrit and hemoglobin, and pretty much would tell you if you're anemic, okay, or if you're actually clotting at a good time. So pretty much these are all going to be part of your first lab exam of your first lab. So please study them. Right now, guys, that is the end of today's discussion on chapter 17 and the blood. I will see you in the next video, which is chapter 20. Have a good day.